Welcome to Ms. Chiao's video lectures. In this unit of genetic mutations, we are going to be covering prokaryotic gene regulation. Here, students will be able to understand prokaryotic and eukaryotic gene regulation works. So we're going to first talk about gene expression. What is gene expression? It is when a gene is used to make a protein. When a gene is used, its phenotype expresses as a protein. And here we have an example of a mitochondrial uncoupling protein. The body has over 50,000 proteins, and of the 50,000 proteins, it is made from over 20,000 genes. Every cell has genes to produce any kind of protein, but does not do that. How come? Well, because every cell differentiates itself due to however many genes it turns it on and off. So different cells have different functions which need specific proteins. So the cell is able to differentiate and be able to select which protein should be turned on for certain cells and which protein should be turned off for making specific proteins. However, not all genes are regulated. Some genes are always on. So that's because they're always needed all the time. So over here, just a quick little review. Where do we get proteins made? It comes from the supercoil of chromosomes, and that segment of DNA that's to code for a protein is called a gene, which contains introns and extrons, for eukaryotic cells. Prokaryotic cells just has RNA. Now, there are two main ways of controlling gene regulation. One are activators, which assist in starting transcription, and it's an element that turns the gene on, whereas the opposite is repressors, an element that turns the gene off. So before transcription happens, which is explained over here on the right hand side, initiation, elongation, termination, has to some way start. So can't get to termination if you can't start it. Prokaryotic gene regulation is much simpler than eukaryotic gene regulation because they came first, they're much more basic, they're simpler, and they're usually regulated by environmental factors which will trigger protein regulation on and off. Related genes for encoding one protein are proximal. So if you have a protein that requires 2 or 3 different kinds of genes to compose it together to form one complete protein, the genes are more likely to be next to each other. And that's what proximal means. Let's go ahead and define an operon. An operon consists of a promoter, an operator, and sections of genes that encode for a protein. The promoter is where the polymerase, RNA polymerase, lands on, and the operator. So general gene expression, what happens is a repressor protein will be on the operator to block transcription. So the operator is a coding section of DNA in which the repressor protein will be on there to block the starting of transcription, most likely RNA polymerase, excuse me. The presence of activators remove the repressor proteins, so activators will go ahead and remove the repressor proteins by binding onto it, changing its shape, therefore losing its ability to bind. Then now the DNA polymerase now has room to bind to the promoter and start transcription. So over here, repressor protein, what happens in this picture, a repressor protein will come and bind onto the operator. You can tell the key lock mechanism that this matches that. Once it's on, a RNA polymerase cannot bind on the promoter because it's blocked by the operator, and it's going from 5' to 3'. And then then a activator will come, bind onto this area of the repressor protein, change the shape of it so it no longer can lock onto the operator. Once it no longer can lock onto the operator, due in this case lactose, the RNA polymerase can go onto the promoter, and once it binds on the promoter, it will transcribe the section of DNA to RNA. And what is the promoter? An area on the gene where the RNA polymerase binds to start transcription. So basically you can think that there are two starting places. The operator, which the repressor binds onto, and the promoter, which RNA polymerase binds onto. So specifically, and this is probably the most common example for gene regulation, is the Lac Operon, which is short for lactose. Lactose is a sugar found in dairy products. The E. coli bacteria needs 3 types of enzymes, LAC-Z, LAC-Y, and LAC-A, to break down the lactose. During the time in which it's turned on, when there's enough lactose, so if the E. coli is in an environment and intakes lactose sugar, and as that concentration goes up, the lactose will bind onto the repressor. Then the repressor will change the shape and not bind to the repressor binding site. Then it leaves room for the RNA polymerase coming in and binding onto the promoter, and then beginning transcription for the lactase enzyme, because there's such a large concentration of lactose, which it needs a lactase enzyme to break it down. So here, let's go ahead and look at the parts. Here, the cap is the promoter, and the P is the repressor. And what happens in the 3 segments of the enzyme, LAC-Z, LAC-Y, and LAC-A. Eventually, once it's transcribed this part here, it will go through splicing via mRNA. So here you have 4 different situations that can happen. Low glucose, which means low sugar. That means it's not dependent on sugar. There is lactose available. When there's enough lactose, the arrows will point to that it will start making lactase. However, if there's high glucose and there's no lactose, this is a test that shows you that this sequence here will only make enzymes for lactase, enzymes for lactose. So this just proves if it's in a high glucose environment, it is not breaking down sugar. However, because there's no lactose, the gene regulation does not happen. Also, gene regulation does not happen based on low levels of glucose, because it doesn't matter, because lactose is unavailable, period. You gotta have the presence of lactose in order for the reaction to happen. Another example is when you have high amounts of glucose with some amounts of lactose, and when that happens, then the gene expression will be turned on because there's the presence of lactose. It hits like a certain threshold of concentration of lactose, and bam, it kicks in and it starts. The last step is that the lactase enzyme will break down lactose, and as lactose concentration amounts decline, I forgot to put in concentration, it should be right there. As lactose concentration amounts decline, it no longer can bind onto the repressor, And now the repressor can bind onto the promoter. So the lactose, as it gets broken down, is no longer attached to the repressor, and the repressor goes back to its original shape and bind onto the promoter. So let's go ahead and do a quick review. You have your binding site gene with the 1, 2, 3 different kinds of enzymes. The regulatory gene, the reason this is called a regulatory gene is the part of the gene that helps regulate whether it turns the gene on or off. And here's the Lac Operon. The operon includes the promoter and the genes that's required. Now, the repressor protein, there's a regulatory gene for that. And then, of course, the 3 different lactose breaking down enzymes for lactose. And the last regulation in which prokaryotes are known for is environmental control. Some genes are turned off due by their own increased concentrations of their own products. So once it reaches a certain amount of concentration that the gene or the cell starts making, when it hits enough, it's like almost like population control. When it hits a certain amount of concentration, it's okay, enough. We don't need that anymore, and it shuts down. And that is the end of prokaryotic gene regulation, and the next lecture will be on eukaryotic.